Okay, in the last video lecture, we talked about mitosis, demonstrated what it was. You'll notice that the background here is onion root cells. Look, what is that? Anaphase. Here's some more anaphase. Way up here is some telophase finishing up. We're starting into cytokinesis here. You might wonder, well, how is it that scientists know what really is happening during these phases of mitosis? It's because they can see them in a microscope. That's how they can know. They actually look at it. They watch it happen. With living cells, they can watch this whole process happen. And that's the answer to that. Let's take a look at some cells here. Maybe I can get out of the way. This is some onion root cells. And just so that you see, again, here it is. Same slide as we were looking at, actually. And there's lining up on the plate and just finishing metaphase and just starting anaphase. Okay, here we go. Here's cytokinesis starting again. We've got the new nuclei forming. So we can look at this. We can look at other cells. Here's some more with a different kind of stain. Here's some more with a different kind of stain. So students can do this in high school. This is real easy to do. And with a decent microscope, you can see the nuclei, and you can actually stain this properly, and you can get up to 400 power and look at the nucleolus inside the nucleus. So these are things that we can actually see. We're not guessing at what happens here. We actually see it. Okay. So now let's talk about cancer. What is cancer? Well, cancer is a growth disorder of cells, and basically it means that normal cells grow uncontrollably, and we get a tumor, a mass of cells that just keep dividing. Sometimes those tumors are invasive, and we call that malignancy. If it's benign, it just grows in a mass in one place, and sometimes we remove it, sometimes we don't have to. But if it's malignant, then it's invading tissue and disrupting the normal function of that tissue. If cells from the malignant tumors metastasize, which means they break off, pass through the blood and get to other areas of the body, then this kind of a tumor is really bad. So we try to control this from using surgery or using different chemicals that stop or inhibit cell reproduction. But the treatments are difficult sometimes. If we catch it early, of course, it's easier to treat. What does it look like? Well, here we go. Here's invasive in a lung. Here's invasive tissue in a lung. How about a diagram of a tumor? You can see that this carcinoma of the lung is now metastasizing, getting into the blood vessels, and these uncontrolled cells are traveling into other parts of the body. There are quite a few good videos that I could share with you. I won't here now, but that actually discuss treatments and so forth for cancers. If you're interested, you can look things up on the internet and find some pretty good things. So as I mentioned before, I'm going to go back to this cancer in a minute and take a look at your lab for the unit, because it's all about cancer too. But I wanted to look at this slide, which is in your book and also in the PowerPoints. because it reiterates what I did mention before. Why do human cells age? We don't know why they age, but I mentioned that after about 50 times that cells double. You know, we're following that DNA, we're following it from one place to the other, from one cell to the next. And so if we look at the population doublings, the number of times a population can double in a cell culture, cell tissue culture, what we see is that at about 50 doublings, the cells just die. Telomerase is causing the divisions. What we think is happening is that when we look at DNA and look at it as it is replicating, there seems to be a tail on the end of it. And every time that DNA replicates, the tail gets shorter. It's like there's a key or something on there that keeps it all alive and functioning. And when those tags, whatever they are, run out, then that cell line, starting with an original cell, ceases to exist. Ceases to exist. Okay, so let's look at the lab. So when we get into the lab, and there is a video that demonstrates this lab, but I want to talk about it with respect to the job of a pathologist. Here we go. We have normal lung tissue here. And the diagrams represent different stages of mitosis. So when we look at the picture and match it up with the picture here, okay, this one's interphase. Then we can label the interphase. And we can go back to the index and choose. We got to find out what this one is, because that's another one we can label. And is it anaphase? No, it's prophase. So we grab the tag, put it in, and check it. All right. So what we do now is we count. Now there's 20 cells. We have one in prophase. So 1 out of 20 is 0.05 or 5%. So our mitotic index is 5%. And this is what a pathologist is going to do. He is going to look at sections of the tissue that was sampled in a biopsy, microtome it into thin sections, and start looking at what stages with his special stains that the cells are in. And he's going to count how many cells are in mitosis versus the number of cells that are not in mitosis, that are in interphase. Okay, so let's compare cancerous lung, and we can see that that one's in mitosis, this one's in mitosis. Do we have any others in mitosis? Well, I don't see any. Let's check it out. So what is this? This is anaphase. Okay. This is anaphase. Okay, and then these are again interphase, right? Is there anything else here besides interphase? I don't see anything. Interphase. Oops. Interphase. Interphase. Okay, check.
Use the cell cycle cards to check your cell labels.
I got something wrong. I must. Oh, this is telophase. That's anaphase. Sorry. Okay, now let's check. Okay, I got it. So we have two of them that are in mitosis. We have one in anaphase, and we have 1 in telophase, so we have 10%. So cancerous tissue has twice the mitotic index as normal lung tissue has. Now, a pathologist knows that when he goes back to these, he has all kind of cells in the body, all kind of different tissues, and he needs to know what is normal for that tissue. Normal stomach cells, for example, are going to have as much mitosis going on. See, there's mitosis. There's mitosis. 2 out of 20, 10%, will have as much mitosis going on as cancerous lung. So you have to do it tissue by tissue, and you have to actually get the data for this lab to understand it and to answer all the questions properly. But different tissues have mitosis going on normally at different rates. So you have to compare apples with apples, and a pathologist is trained to do exactly that, to be able to look under a microscope and recognize not only whether the cell is in mitosis or not, but also whether the cell is a lung cell from lung tissue or stomach cell or whatever. Now, the other important thing is that if a biopsy is taken of a patient and it comes back as possibly cancerous tissue, that a second biopsy is needed, at least a second biopsy is needed, because there is a chance that a particular sample or section, a thin section of a sample, could give erroneous results. That there might be more mitosis going on in that little piece of tissue than is average. We call that normal, right? But it's average. Is normally average. It is not abnormal to be above average or below average. It is normal for things to be average when you compare a whole series of samples. So taking one sample is not enough. We need to take multiple samples sometimes. And so pathologists almost always look at more than one sample. And that's why biopsies generally are taken of multiple tissues in multiple places at the same time. But this is what a pathologist does, and who knows, maybe you will be an assistant to a pathologist sometime in your future, and you'll develop the skills to do this. You'll develop the skills with the microscope, and you'll develop the skills in identifying the different kinds of tissues. Practice, practice, practice is what happens. You may sit at a microscope 5 to 6 hours a day analyzing samples from multiple patients. It's very serious business. It takes a certain skill and aptitude to do this, and it can be quite a rewarding occupation, even though you might think it's boring. If you do this in the future, you might find that you really like it, and it's not so boring. It can be quite interesting once you understand what you're looking at. So that's the application, in part, of this lab, and in the next video lecture, we're going to talk real hard about sex, about what sex really is. And there's some pretty amazing stuff. It is in the discussion of sex, or mitosis, not mitosis, meiosis, that we can make a connection with Charles Darwin and others who have studied genetics in the past. Guess who? Gregor Mendel, right? Gregor Mendel. He was a monk. What is a monk? Do you know? You know what I think I did? I think I forgot to turn